Okay, so today and tomorrow, or yesterday and today, if you're watching this on Gold Day, we are talking about the cytoskeleton. Woo. All right. Yeah, so the cytoskeleton is one of those one of those organelles that, like, in your lower level bio courses, sometimes it's mentioned, sometimes it's not mentioned, sometimes it's just brushed over. But we are going to talk in detail about just how important the cytoskeleton is, starting with functions. This is a general overview of the functions. There are different components that we are going to talk about as well, and those different components have more specific functions. So the first one is mechanical supports. We use them to maintain our shape. This is particularly important for cells that don't have cell walls. So, important for cells without cell walls. So, all of the other cells in our bodies that do not, or our cells in our bodies don't have cell walls because we are not plants. So, all of our cells have these cytoskeletons that help maintain our shape so we're not all flattened out or too wiggly to carry out our functions. So, the cytoskeleton acts essentially like our skeleton does. Our skeleton makes sure that we are not just like a blob of like skin and water. Cytoskeletons do the same thing for cells. Another really important function that cytoskeletons have is movement. Or cell motility. In cell motility, that is both changes in cell location and the movement of cell parts. So when we talk about cell motility, not only does that mean like actual cells like moving around their environment, but also the things that move around within the cells. So we talked about mitochondria being dynamic and being able to move around. That is in part in thanks to the cytoskeleton. Movement generally relies on cytoskeleton and motor proteins. So, generally, because there are always exceptions, requires interactions. If you're a shorthand person, I find that I/A works for interactions. Interactions between the cytoskeleton and motor proteins. So our cells wouldn't be able to move, and things in our cells wouldn't be able to move if it weren't for both the cytoskeleton and motor proteins. For example, cytoskeletal elements and motor proteins work together with the plasma membrane to move along fibers outside the cell. So, say we've got a cell that's sitting in its little aqueous environment. There are fibers running in and out of that aqueous environment. We will talk about those when we talk about the extracellular matrix. So all those fibers that exist outside the cell, motor proteins and cytoskeletal elements inside the cell can help move the cell along those fibers like train on train tracks. So that's just one example of cell motility. There are 3 major components to the cytoskeleton. 3. 1 we're going to talk about today, and 2 you are going to talk about tomorrow. So 3 major components. The first one, the one that we're going to talk about today, are microtubules. The second is microfilaments. And the third are intermediate filaments. So microtubules, microfilaments, and intermediate filaments all work together to comprise, I guess is the word, the cytoskeleton. Today we are just focusing on microtubules. So microtubules are the largest component of the cytoskeleton. The microfilaments are the smallest, and the intermediate filaments are kind of in between. So, microtubules. Microtubules are hollow rods constructed of globular proteins. Globular, fun word to say, proteins called tubulins. So tubulins are a type of protein that come together to create microtubules. Tubulins are dimers, which means they have 2 subunits. So when we talked about quaternary structure with protein structure, a dimer is one that consists of 2 polypeptides put together, and tubulins are an example of one of those dimers. So tubulins are dimers, which means they have 2 subunits. Those subunits are called alpha tubulin. That's not an alpha. And beta tubulin. They have plus and minus ends. This does not denote charge. The plus and minus ends essentially tell us where they grow from. So they elongate at the plus end and dissolve at the minus end. So this is our microtubule. We'll get into structure later. If this is the plus end and this is the minus end, essentially it would grow from the plus end and shrink from the minus end. Does that make sense? All right, cool. So dimers, which means they're made of two subunits or two polypeptides because they are proteins. They are named alpha and beta. A lot of the times, whenever we have dimers, they are called alpha and beta. Hemoglobin is another example. So hemoglobin is made up of two subunits. They are called alpha hemoglobin and beta hemoglobin. Just helps us figure it out a little bit better than if it were A and B tubulin. So their main function is to shape and support cells, and to serve as tracks along which organelles with motor proteins can move. So vesicles, right, are our little like transport vehicles that get stuff where it needs to go within the cell. Those vesicles have a lot of motor proteins on the outside of them, on their membranes. Those motor proteins get sent on a track of microtubules, and those motor proteins take that vesicle from one part of the cell to another, lying across those tracks. So that helps not only the cell stay organized, but also helps the Golgi get those vesicles where they need to be. So, like I said, they guide vesicles from ER to Golgi. So, you know how the Golgi has those trackers that tell stuff where to go. The ER doesn't necessarily have that, so it relies on microtubules to make sure that our proteins get from the ER to the Golgi. They're also really good or involved in separation or anaphase in cell division. So if you remember those pictures from when you learned mitosis, how the cells are kind of pulled apart, it is the microtubules that allow that to happen. They are kind of the tracks along which everything gets pulled from one side to the other. So far, so good? Cool. There are two specific types of microtubules that I'm going to talk about. The first being centrosomes and centrioles. Centrosomes and centrioles. These are in animal cells. In animal cells. Microtubules grow out of the centrosome. Which is just a region often located near the nucleus. That is not how you spell near. Goodness gracious. I didn't say not on Monday, but almost Monday. All right. Often near the nucleus. So these some microtubules have to come from somewhere. Specifically in animal cells, that somewhere is called the centrosome. Within the centrosome, are a pair of centrioles. Centrioles are nine sets of triplet microtubules in a ring. So, if we've got 9 microtubules in a ring, 1, 2, 3, 4, 5, 6, 7, 8, 9, and each of them is a triplet, that is what a centriole looks like. These help in microtubule assembly, keeping things organized, monitoring the pluses and minus ends. And this is only in animal cells. So some eukaryotes with centrosomes and centrioles, or without centrioles and centrioles, organize microtubules in another way. This is just how animal cells do it. So if you think about, you have to write this part down, but if you think about how, when we talk about cell division, we've got those little like tic-tac looking things that pull from one side to the other. Those are our centrioles and centromeres. They monitor how our microtubules grow and kind of span across. Okay. We cool with this? All right. For some more fun stuff, we are going to talk about cilia and flagella. Really cool parts of the cytoskeleton. So, cilia and flagella are both microtubule containing extensions that project from the cell. Oh, I don't know how that worked. So, cilia and flagella are our little extensions of the cell membrane that mostly guide in movement, that contain microtubules. So, many unicellular eukaryotes are propelled through water by cilia and flagella. And I'm going to abbreviate that. So, essentially, if a unicellular eukaryote wants to move through water one way or another, they will have these cilia and flagella attachments that allow them to do that. But when cells are held in place, cilia and flagella are used to move fluid. For example, The lining of your trachea in your throat have a lot of cilia. So lining of trachea equals cilia to move mucus. So when you cough and all that junk comes up, that is because your cilia are helping to move fluid one way or the other. Another example, I think is a really cool example. So your oviducts or fallopian tubes have cilia that move eggs towards the uterus. So when that egg moves down the fallopian tube, whether or not it is fertilized, it is the cilia that line that tube that helps it move from one place to another, which I think is really super cool. So I have a quick comparison and contrast chart. So we've got specifically motile cilia and our flagella. So in this specific chart, it's not necessarily those ones that are held in place that move fluid. We're just talking about the ones that help us move around. So when we use cilia to move, we usually have really large numbers. When we use flagella to move, we usually only have 1 or 2 per cell. So if a cell is moving through water via cilia, we're going to have a bunch. Meanwhile, if we have a flagella, odds are it's just going to have 1 or 2. The motion in which they move cells is also really different. So cilia are like oars. If you think about how you move a boat. So we've got alternating power and recovery strokes. Like oars of a boat. So if you think about the movement that is rowing, right? You've got the one that's in the water that's going to push you, and then you've got the one to go back to the front. That's where the oar is not necessarily in the water, so it's not doing anything. It's just getting you back to normal. That's the way cilia work. So all these little cilia work like oars to help project the cell through whatever fluid it is in. Cilia have an undulating motion. Undulation. Like the tail of a fish. So while we've got our like boat or cilia, our flagella just kind of move us like a fish, or like if you're in the pool and playing mermaids, you just kind of wiggle. That's how flagella work. One more thing about cilia. Cilia have another function, and they can essentially act like an antenna. So signal receiving antenna. The antennae are non-motile, which means they don't row. And usually there's only one. and that is called the primary cilia. So if a cell only has one cilia, most likely it is used for signaling rather than movement. I forgot to close that parenthesis. This is really crucial to brain function. So if we have my little brain cell here, these guys down here would be our like signal receiving cilia. So really important to brain function, also really important to embryonic development. I took an entire course over development. Specifically, I think we studied worms, flies, and people. And a lot of development is based off of signals that are received from one cell to another, and a lot of concentration gradients. So these are really, really important for embryonic development, making sure that cells wind up specializing the right way. I have one more little thing for us, and that is the similarities in structure. So and this is with cilia and flagella. They are both groups of microtubules that are wrapped in extensions of the plasma membrane. So they're both a bunch of microtubules wrapped in cell membranes. They are also, there we go, sheathed, or you could also say wrapped, doesn't matter. They are both anchored to the cell by something called a basal body. Basal bodies are shaped like a centriole. In fact, the basal body of a sperm cell, right? So think about sperm cells, they have that little tail, that basal body where it's connected to the cell, will actually become a centriole in the fertilized egg. Which I think is really cool. Dual purpose. When they bend, they're usually due to motor proteins called dyneins, which are attached along each outer microtubule doublet. So, might get a little complicated. So, attached along each outer microtubule doublet, i.e., typical dyneins have feet that walk along a microtubule and this uses ATP. It's not just doing it on its own. Using ATP, and this causes the microtubules to bend. So let me see if I can find a video really quick, because videos make everything better. Where am I at? Aha. So this right here. We've got its little feet that are moving. This one is specifically moving a cell along a microtubule, but if like the top of this guy was attached to one microtubule, and so we've got two microtubules together, if it moves one, it can bend the entire organelle. So let's see. If we were another microtubule right here instead of this cell, if this one was staying in place, but it were walking this way, it would essentially cause the entire thing to move this way and bend. Does that make sense? A little bit? All right. So every time it takes a step, it is because a molecule of ATP has been hydrolyzed. So each step is an ATP molecule, which is why our cells take so much energy. So, 1 second. There is not an exit ticket for today. So, there's that.